actually just going to be talking about Diamondback moth and brassicas. There are a lot of other pests in brassicas, but Diamondback moth is really the one that gives us a problem. So I want to just start off talking a little bit about the treatment interval approach to managing insecticide resistance in Diamondback moth. Diamondback moth is a problem in Florida and globally because it develops resistance to insecticides. When you apply just about any insecticide, you can assume that a certain proportion of the population in your field may have tolerance to that. Some females may survive, will produce offspring, and will pass on that genetic tolerance to the second generation. So what you don't want to do is treat successive generations with the same modes of action. So we use a 30-day treatment interval, which corresponds roughly to the generation time of the Diamondback moth. They spend about a week as eggs, a couple weeks in the larval stage, and about a week in the pupal stage. And you'll actually see a 30-day treatment interval on some of the labels that you can apply the same modes of action more than once within that 30-day treatment, but then in the next 30-day interval, you need to use different modes of action. So here in this schematic, I've started off with the early establishment of a cabbage crop and broken it up into three 30-day intervals. You don't necessarily have to start applying insecticides as soon as the cabbage or other brassica crop is in the ground. If you're growing brassicas in Florida in the fall, you may not have problems with Diamondback until a few weeks into the crop, and you might not be applying until you're in these early, say early cupping stages. And you might want to go ahead, say with some of the better insecticides that we've evaluated over the past years, you might want to go out with Proclaim, Radiant, Zentari. And if you apply them in this what would be this mode of action group in the middle here, that's fine. But then when you get to the last mode of action group, as we're approaching harvest, you wanted to use completely different insecticides such as Torac, Dibrom, and Avant. Now, if you're growing brassicas in the spring, when we consistently have high Diamondback moth pressure, you might want to start off with an at-plant treatment of a diamide, say Coragen or Verimark, because they're systemic and will have that extended effect. Since it is systemic, you probably only want to apply a diamide once. Whether you apply it once or twice in that first treatment interval, you don't want to apply it again in successive treatment intervals. So that's the way that concept works. Another thing that we want to keep in mind is the relationship between the crop phenology and the insecticides. When I started working 5 years ago with Diamondback, I assumed the diamides were the most effective insecticides for Diamondback. We're talking about Coragen, Verimark, XRL, Arvanta, because they have this systemic action. They're really the only systemic insecticides that are effective against Diamondback. Movento has Diamondback on the label. It's the only caterpillar on the Movento label. I have not worked very much with Movento, but really when we're talking about systemics, particularly with something that can be applied at plant to the root system, we're talking about the diamides. So the question that I was wondering, growers were wondering, early on, when do we want to take best advantage of that systemic action? Is it early on? Because when the female goes into that growing point, you see in that early plant, lays an egg, the larvae carry out feeding on that growing tip of the plant, it's destroyed and nothing is going to be harvested. So is that when we want to use our best materials? Or do we want to use them later on when the head is starting to form and when a contact or even a translaminar material might not really penetrate to where we want to get? So this is a question that we're going to get back to a little bit later, the optimal placement of diamides. And just to be clear, the diamides are the group 28 insecticides. They're ryanodine receptor agonists, Coragen, Verimark and XRL are the same active ingredient, cyanotroniliprole. Arvanta is really only can be applied to the foliage. And I've listed some of the combined products down there that have diamides in them mixed with other modes of action. When I started working with folks at the University of Georgia in about 2018, we carried out a maximum dose bioassay that was developed by Stormy Sparks and David Riley, in which we used the top labeled rate of a range of insecticides against field collected populations to try to clarify patterns of resistance in Florida and Georgia. And this is a publication that we put out in 2020, which I'd be happy to send anyone who's interested. And here's a figure from that publication showing some patterns for chlorantraniliprole on the left, which is Coragen, cyanotroniliprole on the right. at least as I'm facing it. And really what I wanted to show you with this map is the main Brassica area growing areas where I have been working. So you can see up St. Tri-County area, up St. John's, Flagler, Putnam, down where I am here in Hillsborough, Manatee. And we've also collected and tested from a variety of farms around Lake Okeechobee. So it's a pretty simple test. We would just mix up the top labeled rate in the equivalent of 30 gallons per acre. We would treat a cabbage disc. We would weigh the disc because we're also getting information on feeding. Then you can see we would take second or third instar Diamondback from somebody. We would get populations from the field, rear them through the next generation in the greenhouse, place them on that disc, and then give them 72 hours to feed. And after 72 hours, we would look at mortality, and we would also see how much feeding had been done. And this would be replicated 4 times. So this is a table that I presented a couple of years ago, or started presenting last year, at least maybe at the Citrus Expo. And don't normally like efficacy tables, but we've worked enough on this that I'm comfortable with this. And you'll see, I'll show you some of the populations that are behind this table, basically. And these are patterns we've seen in Florida and in Georgia. We've consistently seen good results with Proclaim, Radiant, Zentari, have looked less at Diabrome, but good results. Torac, I just moved from the Citrus Agro Expo meeting a couple of days ago up to the high group from the intermediate group, and you'll see in some of the populations it does perform consistently rather well. The ones in green and the percentage mortality there, when it says above 74% or 71 to 61%, that's from the paper with David Riley. With Harvanta, with Veromark XRO, we've had variable results, as you will see. Dipel is fine. We've consistently seen better results with Zentari, and there are a lot of other BTs, but those are the only 2 I've looked at. Avant, you will see, is never the best material. It is also never the worst material. So Avant, I think, is a good rotational partner for Diamondback. With Coragen, you're going to see quite variable results. Initially, looking at Lanate did not work well. Same with the pyrethroids, which we, on the whole, do not recommend for Diamondback. I was involved in generating some of the data for NAC a number of years ago. We do have a special label for NAC for Diamondback, but it has never really stood out as a Diamondback material, and neither has Ryman. And I think my screen's a little concealed here, but Intrepid should be down there on the bottom. Reached out to Dow about Intrepid years ago when we started doing this, when there still was a Dow, and they were very clear on the fact that Intrepid is not particularly good Diamondback moth material, that Radiant was their good Diamondback material. So I want to show you some representative populations going back from the past 4 years of this maximum dose assay that we've done here. And we collect populations, or our collaborators have helped us collect them. Glades Crop Care has been particularly helpful in this. And we bring them back, we rear through to the second generation, and we test those second generation larvae in the lab. And here are just 2 populations from Hardee County, and you can see the percentage survival after 72 hours. The blue column is the control. Usually want to see that over 80% to feel confident in the test. So what I want you to see are those very tall yellow columns of Lanate. We consistently saw Lanate really not doing much of anything. The red column there is Brigade. This is the main. We looked at Brigade and I think Warrior, but the pyrethroids were consistently not very good. You can see Dipel in there, Intrepid, also not very effective. We really stopped looking at Intrepid after this year. Coragen is the light green column in the middle, and this is the sort of pattern we started to see pretty early on, that it was, we've got 50% survival, and these larvae are stuck in a petri dish with nowhere to go and nothing else to feed on, and yet half of them are still alive 3 days later. And as you go looking towards the right, what you're going to consistently see, we have a bright blue for Proclaim, we have pale sort of gray for Zentari, black for Radiant. Those products are consistently on the right where things are doing an effective job. And in this particular season, we did get good results from Harvanta on these populations, and variable with XRL. So here are some populations from 2020. Again, we couldn't test everything all the time, but focusing on the diamides, seeing not particularly good control with that for that Charlotte County population. If you look at the transplant production, this is from a large-scale transplant production house in Sumter County, and you can see there was complete failure of the 3 diamides. There were actually more Diamondback surviving where we treated them with Coragen than where we didn't do anything. Hervanta, Exerel, so real complete failure of all diamides. And we would see this, and actually we're advising people really against using the diamides just because this was a pattern. But what is also interesting, you'll see when the diamides aren't really working, you're looking over to the right, you're seeing good results, Radiant, Proclaim, Zentari, and with some of these other materials. Some more populations from 2020, poor performance of diamides, or again, from that Charlotte County, a different farm. These are all commercial farms, complete failure of the diamides, reasonable results with some of the other materials. And one last population where the diamides did very little, but Proclaim, Radiant, and Torac killed everything. So I want to switch gears a little bit and talk about some work we've been doing to starting back in 2018, trying to understand the best placement of these diamides back when it seemed that they were more of the optimal products. And a grower here in Manatee County was knifing Coragen and Verimark in a few weeks after crop establishment as a way to have the product be taken up by the roots, because it is, of course, systemic. Now, there was some talk in a previous presentation about cabbage under plastic. Most cabbage is grown bare ground. So if it were grown under plastic with drip, you would have better luck, at least delivering the diamides to the root system. But I was wondering, is this actually an effective way to deliver Coragen or Verimark to the root system? And as it turns out, DuPont, back when there was a DuPont, they did this work. And what I want you to look at in this slide, which was provided by DuPont, in those squares, what you're looking at is the movement from a drip irrigation. The little blue circle is the drip tube, and they collected data on the movement over 14 hours of Vidate and AdmirePro and Coragen. Now, Vidate and AdmirePro, imidacloprid, like a lot of the neonicotinoids, is quite these products are quite mobile in soil. But the point that I want to make with this is that the diamides are really not very mobile in the soil, and this has already been proven by the manufacturers. So if you're thinking of knifing Verimark or Coragen in with the hope that it's going to move with that gradient towards the root, it's really not going to work. These products, the diamides, are only effective if they were really delivered directly to the root. And in recognition of that, the cyanotronilprole Verimark actually has a nursery application label. It can be applied in the nursery up to 72 hours before being shipped out or going into the field. This way, here you've got a picture of it being applied with a boom. In theory, a grower could do this. We've done it on a small scale to evaluate this to see, is this way of applying cyanotronilprole Verimark effective in terms of giving a longer term residual efficacy as opposed to, say, doing it at planting or foliar. This is some work done by a master student, Esnai Muntali. You can see her picture there, graduating last month. And what we looked at, and we did this actually a number of times going back to 2020. I'm going to show you the last 2 times we did it, which I think were the most, we weren't disrupted by COVID or anything like that. But we have looked at this a number of times, and we looked at 8 different treatments, or 7, including the untreated control. So what we looked at, we treated cabbage with Verimark in the tray. We treated cabbage with Verimark or Coragen at planting. And we had foliar applications of either Exerel or Coragen 3 weeks after planting, and either Exerel or Coragen 5 weeks after planting. So each plot just got one of these treatments. And then what we did is we took the plant tissue and we fed it to a highly susceptible lab colony to see how long those caterpillars would die. The logic being, If they die, it's because the insecticide is still there. And if they don't die, it's because it's not there. And we used a lab colony, unlike the data I showed you before, which was from field colonies, which that wouldn't have worked because there's a lot of resistance in the field. So again, just to emphasize the way we did it, you can see the tray treatment. You can see us ladling the at-plant treatment, which is a lot more precise than in terms of the amount than what you might get with a drip, an actual drip application, but we're trying to be precise. And you can see there were 2 foliar applications, one at 3 weeks, one at 5 weeks, in which the plants received either XRL or Coragen. And to sample, initially we would take the whole plant because there's not a whole lot there when we would provide that to the caterpillars. And just got some images here. We were always trying to be consistent and sample that innermost sort of tender growing central part of the plant where the female diamondback at least most like to go in and lay eggs, and where you really consistently find those early infestations on the plant. And as I said, we used a highly susceptible colony. It was from Delaware. We got the eggs a week before, as and I would rear them out on plants in the greenhouse. And the following week, when we had those second or third instars, we would put them out and do the test. And we'd look at 72 hours to see if they were alive or dead. And we also took information on feeding, with very striking differences with some of these treatments about the amount of feeding. So we took data every week for 10 weeks, but I'm going to pool some of those data just for simplicity's sake. So what we're looking at here is survival after 72 hours from the 2 runs of this experiment, from fall 2021 and spring 2022. And what we're looking at is the number of larvae that survived after 72 hours. And we've got the control, where everything was pretty much fine. You can see Cyanotronilipol tray, which is the Verimark in the tray. You've got Cyanotronilipol plant, which is Verimarket plant, and you've got the Coragen plant. So you can see that the tray and the at-plant treatments pretty much killed everything those first 3 weeks. A little bit of difference the second year with the Verimark, but they pretty much killed everything. And then at week 3, you can see we applied these foliars. We applied Coragen in some plots and XRL in other plots. And what we're looking at here is the two weeks after that application, averaging. So weeks 4 and weeks 5, we collected it. I'm just showing you the average of those 2 weeks. And you can see that everything's doing better than the untreated. There's continuing to be a residual effect, but the tray treatment is at this point no different from the foliars, which were not as good as the ones that were applied at plant. So that's kind of striking that we're continuing to get the suppression from the at-plant treatments. Lastly, we applied Coragen or XRL in a different set of plots 5 weeks after, and that's what we're looking at here. So we applied those last ones week 5, we collected samples week 6, 7, 8, 9, and 10, and that's what we're looking at here, the average of 5 weeks, the last 5 weeks, pulling it all together. And again, I should say when I started this, I assumed that a few weeks after these treatments were applied that the material would break down, it would become too dilute in the plant, and things would start looking like the untreated. Nothing ever approached the untreated. It's some of the very last weeks with some of these, if you looked at it week by week. But we saw much longer residual efficacy than we expected from all of them, and a much greater impact. I mean, you can see those 2 at-plant treatments were still 10 weeks after application. We're still killing a lot of these, most of these larvae. And remember, this is a highly susceptible lab colony. I'm not saying you're going to see this sort of control in the field, rather the opposite. So what we're seeing, and I am assuming this is analogous to diamides not being mobile in the soils, we're seeing, at least in cabbage, these materials being retained, that we're seeing a much longer residual effect than I was expecting. And I'm assuming that for a field population, these are sublethal doses. So what you're kind of doing is feeding sublethal doses of diamides to field populations, which to me helps explain why we see these problems, why we see this resistance. It's just retained in the plant for much longer than was expected. And we sent some to a lab in Canada for analysis, those at-plant treatments. It's very expensive. I was only able to send the at-plant ones, but indeed 10 weeks after planting, they still were detecting diamides. So something else just to think about is the feeding. We're taking data on feeding, and I'm going to pool the 2 years just for simplicity's sake. So going back to that first 3-week interval, you'll remember all the untreated were alive and pretty much everything else was dead. Well, they fed quite a bit before those 72 hours. So not much of a difference amongst treatments, nor much of a difference between the treated and the untreated. I mean, it is statistical. There was statistically less feeding, but still not a huge difference, indicating that they do feed quite a bit before they die. And we're seeing the same thing. This is that next interval. We were looking at weeks 4 and 5, where we've got those 3 foliars. None of these treatments are really separating statistically in terms of the amount of feeding suppression, whether they were applied at plant or much later. There aren't huge differences with the untreated. And that last group, when we're looking at the last month of the crop leading up to harvest, the lowest really is still the Verimark at plant in terms of reducing feed-in. It's not statistically different from the Coragen at plant or the 5-week XRL. And one of these, the Coragen 3-week, was not statistically different from the untreated, but it's important. We usually just think about mortality and what is killing the caterpillars, but with a lot of these different treatments, we've seen quite a bit of feeding before they died. So in wrapping up, I want to go back to the maximum dose studies that we were doing again this spring. And I just want to say that we actually saw a slightly different pattern with some populations compared to what I initially showed you from those first few years. This is some of Amanda Faruya's master's work. Amanda looked at diatomaceous earth alone and in combination with the BTs, that is in combination with dipel and with Zentari. A grower reached out to me a number of years ago, and I was finally able to look at this. I mean, so the diatomaceous earth is the gray column. If you look at the cabbage from Martin County, there's only 40% survival, which was kind of promising, but more often than not, what we saw was what you see under the Palm Beach County population, which is a little bit of something happening, but not really very much. Diatomaceous earth is normally used as a stored product material. It abrades the outer, the cuticle of the insect and causes it to dry out. But we looked at this as a feeding test, and in a spray tower, did not see really promising results either alone or in combination with the BTs. But the other thing that you can see by looking at these figures is not very tall columns. We started looking at lanate again. That first graph I showed you from 2019, lanate was 80% survival. Brigade was 80% survival. But look, these populations were actually pretty much susceptible to everything, including lanate, including pyrethroids. You can see all of the diamides worked reasonably well or quite well. So you can see we looked at Spearlep combined with Leprotec a number of times this season, because there's been a lot of curiosity about that product. And, and you can see that in some of these, it worked reasonably well, that combination of the two. So I just wanted to say we have seen some more susceptible populations, and this is corroborated by the good people of Glades Crop Care who submitted a lot of these. They were also saying, yeah, a lot of these weren't that hard to manage. However, we are also still seeing populations that do have pretty significant tolerance to the diamides. This is also from the spring. You can see the gray columns there, the diatomaceous earth next to the untreated, not really doing much of anything. But you can see XRL and Coragen. We did not look at Harvanta, but XRL and Coragen not working particularly well or at all in these two populations. But some of these other products, as we'd seen previously, Proclaim, Zentari, Radiant, Torac, Dibrome, working quite well on populations that are tolerant or resistant to the diamides. So yeah, that's all I wanted to say. I wanted to thank the funding sources for this work, and also thank the people from Glades Crop Care, as well as the people in the lab who helped do the work.